Hey everybody, welcome to lesson 16. It's the final lesson of this class, so congratulations for making it so far. And I hope this is a really interesting lesson to cap off the entire class. We're going to pull in a lot of different information from all the previous lessons, so if anything's confusing, as always, reach out to me. I'm happy to clarify anything as it needs to be clarified. So, in this lesson, we're going to be talking about disorders of the immune system. So, microbiology as a whole discusses the micro world, and usually that is, fungus and bacteria and viruses, and then immune cells. And this kind of brings together how all of those interact and really impact our day-to-day lives because more and more evidence has surfaced that has shown that things like cancer and autoimmune diseases are caused by microbial organisms and our body, our immune system cells reacting not necessarily poorly, but not reacting perfectly to these kinds of attacks that come from the outside. Now, a brief review, your immune system is designed to ultimately protect you, but it can have misfire. So there's a lot of different systems in place to make it so you don't go and attack your own tissues, you don't go and attack your joints, and you don't, not respond to an invader, an invading microbe, because that would be equally as bad. You need to respond. But we do this every single day millions of times a day because there's millions of microbes all up in our business. And every now and again, we're going to mess up. It's just going to happen. And, you might get one cancerous cell, let's say, because it's infected with virus. And usually your body clears that and you're fine. But sometimes, it just is chance over time. There might be a virus that infects you. That cancerous cell doesn't get caught by your body, and then it turns into a mass. And now a cancer has appeared. So how do we deal with that? How do we break all this down? How do we discuss these things clinically? And ultimately, how do we find solutions to these disorders of our immune system? First way we're going to break it down is into a couple different main categories of the types of disorders we can have. So one's hypersensitivity, then there's classical autoimmune disorders, graft and transplant rejections, and immunodeficiencies. There's four types that we're bringing into here, but realistically, like everything else in biology, there might be some overlap, and depending on who you're talking to and in what context, they might shift these, big piles of different disorders a little bit into different categories. But this is how we're breaking it down. So, starting with hypersensitivities, hypersensitivities are reactions that occur when the immune system causes damage instead of protection. So, this is when the immune system is causing damage, and it might be causing this because it is being overprotective, or it might be causing this because it thinks it's being protective, but it's really not. So there's multiple triggers for this, but just know that hypersensitivity reactions are when your body is having a reaction that's actually causing you damage. Type 1 is an immediate reaction, and this is something like an allergic reaction. So this is when your IgE antibodies, so IgE, big E like elephant, those guys usually are going to attack parasites, but we know they also are involved in allergies. So you might have a peanut allergy, for instance. You eat a peanut, a bunch of IgGs freak out, they attach to a peanut protein, you go into anaphylaxis. That is an immediate type 1 allergic reaction. And this comes from exposures to allergens like pollens, food, bee venom is one of them, any venom, really. And you can be allergic to literally anything. Realistically, it's usually going to be pollens, foods, venoms, things that your body sees, has a big reaction to, or sees over time and has an increasing reaction to. And that's why pollen is one thing that you become allergic to because you're just kind of bombarded with it constantly. And it looks a little bit like a thing that should be, taken care of and disposed of versus a nothing. We use a lot of what we call inert substances in things like medical devices, but you can become allergic to those inert substances. Just because you typically aren't doesn't mean you can't be. I have a friend who's allergic to, I believe it's red dye 5, and you can become allergic to things like that. It's something that, might be semi-ubiquitous, but you're allergic to it and you have to change your entire life due to that allergy. There's something called usually it's referred to as a red meat allergy. I believe it's an alpha gal allergy. I would have to double check to be sure, but you can develop this allergy due to a tick bite. So the tick bites you, it has some microbe in it that infects you, and then your body recognizes that but confuses it with this protein in red meat. So then you're now allergic to red meat. It's kind of crazy how these things work. So that's a type 1 hypersensitivity. A type 2 or cytotoxic hypersensitivity is going to be when your antibodies target antigens on your cells, leading to lysis. So cytotoxic shock is generally what this is referred to. Usually it's going to occur when you have some sort of toxin in your body or some sort of major exposure. So those cells start, having misexpression of things. They're kind of deregulated, and your body says, this is going to turn into cancer. We need to kill these cells. This can also happen when you have a really, really bad viral infection. That's just this ramp of a ton of infectious particles that are infecting all your cells. Your body's killing all of them, and you can go into cytotoxic shock from this. One of the main examples, though, is actually a hemolytic transfusion reaction. This is when you have the wrong blood type given to you in the hospital. And this is why blood typing is so important. So if you have a blood type, like say you're AB positive, or actually, let's just say you're B positive, because I believe AB positive is a universal acceptor. So you're B positive and you're given A negative blood. Well, you're going to react to that A negative blood. So that leads to you have a bunch of foreign antigen as far as your body's concerned inside you. We need to destroy all that. We need to take it up. We need to pull it apart. We need to get rid of it. And that leads to this widespread cytotoxic shock that if you're not in a hospital in a controlled environment like that, you're going to most likely die. Cytotoxic shock is very, very dangerous. Now, type 3 is called an immune complex mediated hypersensitivity. This is when you have antibodies clumping up with a bunch of different antigens and it forms these insoluble complexes that get deposited in tissue. This isn't nearly as dangerous as the first two. It can happen a lot with things like cancer therapy. So you have immunotherapies. Remember, those are going to be usually IgG, as in gorilla, antibodies, and those will go in, attack the cancer explodes. It creates these big clumps of antibody because you've been given so much, and there's so much antigen for it to take care of. And then those become insoluble little blobs, and it goes to your kidneys, and it can cause things like kidney failure in the worst case scenario. So you get renal failure. Generally, you're going to be okay. But the other thing that can happen is over long periods of time, you can have increasing issues from an autoimmune sort of perspective. And this is where, again, the two things kind of overlap. So rheumatoid arthritis is when you have antibodies attacking your joints and they're creating these deposits of antibody antigens and in your joints and it hurts a lot and it sucks, right? But that also falls kind of into autoimmune disorders, which we'll talk about later. The last type we're going to talk about in hypersensitivities is type 4 delayed cell mediated hypersensitivity. So, this is when T cells respond abnormally to antigens, causing localized damage. This is usually what you see when someone has really bad poison ivy. So, your body says, whoa, there's toxins here. This looks bad. We need to start killing cells. It's not supposed to happen. You're not supposed to start killing cells and it doesn't happen immediately. It happens a little while later due to signaling errors, essentially, due to a game of telephone that's being told that's telling the T cell this is bad. And the T cell goes, I was told it's bad. I'm going to kill this cell that's exposed to this toxin, even though the cell would maybe be fine at the end of the day. So you have this delayed T cell mediated killing of otherwise healthy cells that have just been exposed to something. And that's the type 4 delayed cell-mediated hypersensitivity. Now we're going to move into autoimmune disorders. So, autoimmune disorders are when the immune system mistakes self-antigens for foreign antigens. And this is why rheumatoid arthritis, even though it's immune complex mediated hypersensitivity-ish, falls actually under autoimmune disorders, because this is you mistaking your self-antigens for foreign antigens, because you're attacking your own cells, you're attacking your own proteins in rheumatoid arthritis. Now, there's not subclassifications of this because there's just too many different types of autoimmune disorders. It's hard, no one really agrees on how to categorize them. So they kind of clinically get lumped into rheumatoid. So that is a lot of different things, but kind of, joints and immune, and then into immune, or if it has to do with a specific organ system like brain, it'll fall into neuro. And if it's heart, it falls into cardiology. So there's not a good way we categorize these autoimmune disorders right now. So when we talk about them, we kind of just have to think about why do they occur and how do we deal with this after it has occurred. And there's really two ways that they occur. One is you have genetic predisposition and then an environmental trigger, such as viral infections. And this is becoming more and more commonly accepted that this is the main trigger for autoimmune diseases. So when we look at something like multiple sclerosis, where your body is actually attacking your brain and causing the slow degeneration over time of all your faculties. This is heavily, heavily linked to Epstein-Barr virus. This is actually something I work on, and it is widely accepted now that Epstein-Barr virus and maybe other viruses cause multiple sclerosis. And that is the trigger, along with these genetic predispositions, that lead you to developing it. We know, for instance, I think it's around 2.5 times more likely to get MS if you have a first degree relative with MS. So that's someone like a brother or a sister or a parent or a child. You are more likely to develop it. So there's obviously something genetic going on, but there's not one genetic thing. So genetic predisposition plus environmental trigger equals autoimmune disorder is the most common way. The other way is you have errors during T cell maturation or B cell maturation that allow self-reactive T cells and B cells. Now, your T cells from your thymus go through a lot of stages of being exposed to self antigens. And saying, "Hey, do you recognize this? If you recognize this, you gotta explode." And then it recognizes it and explodes. But sometimes a T cell goes through that whole system of seeing all those antigens and saying, "I'm fine, I'm fine, I'm fine." Finds one, says, "That's dangerous," and then is told, "You need to explode." And it goes, "No, I'm not gonna." And then it gets out into your body, and all of a sudden you have an autoimmune disease. That's the other way it can occur. The same thing with B cells. So something like rheumatoid arthritis is a lot of B cells creating antibodies against your joints and different proteins and cells in your joints. And that occurs because the B cell goes, I saw something that looks like this. Now I think this is also that thing. So I'm going to produce antibodies against it." And then you have rheumatoid arthritis. So those are the two ways that it can occur. Then there is graft and transplant rejection. So this gets a little bit complicated because it is something that is technically foreign that we're trying to get the body to not reject, and gets into how do we manipulate the immune system to allow us to do things that let people live, like getting heart transplants, without killing them from the transplant itself. So there's four types of transplants slash grafts. Remember, a graft is going to be a small part of organ system. So this would be something like a skin graft usually, and a transplant is going to be a whole organ. So something like a heart transplant. We have autografts, which are from the same individual to the same individual, and this is no risk whatsoever. So if you get a really bad burn on your arm because spilled a bunch of, hot oil on your arm and you need a graft, they might take some skin off your leg, put that onto your arm, and then that acts as a autograft for yourself. There is something else called an isograft, which comes from an identical twin. So this is very, very low risk, but it still holds some risk because remember, we have the same genes in an identical twin, but over time, your body makes different choices. You get different things happening with expression of genes and things like that being upregulated or downregulated. So there is still some risk, but it's very, very low. Then there's an allograft. This is from one person to another person. It's very common, but carries a rejection risk. So if you need a kidney transplant or a heart transplant, the best thing is to find a relative because you're mostly the same. But it might be a random person that you've never met before that is giving you this organ because they're an organ donor and you're getting this organ and they do the best to match it to you, but there's still some different, markers that are off. So you have to be on these immune dampening medications, these immunosuppressants for basically the rest of your life because this organ is different from you. And at a certain point, what actually happens and is really interesting is the tissue in the organ cycles over and it becomes your own cells. So it happens slowly. It happens over a long period, but at a certain point, it should be mostly your organ, which is really cool. But at the same time, you have to be on those immunosuppressants a long time, which isn't great, but you need an organ, so you're going to do it anyway. And that's why it has some rejection risk, but with modern medicine, we're actually able to do this pretty confidently. The next is a xenograph. So recently in the news, there was a case of, I believe it's a genetically engineered pig's heart that was transplanted into someone and lasted for, I think it was 61 days. And that was just the timeline. The person didn't die as far as I'm aware of or anything like that. It was just the timeline that they wanted to try this out because there was no other options. So they gave them the heart and they're, I believe, putting them on either getting them a human heart or doing some sort of other artificial heart now. But these xenografts are really interesting because we have animals like pigs that can be genetically engineered to be more and more similar, at least as far as the heart tissue goes, to our heart tissue and carry markers that are more and more like our markers. So it's very high risk, but for a system which relies on people having accidents or something happening and being organ donors and matching up to other people, this is really probably the only way we're going to be able to support everyone who needs organ transplants is these xenografts and using animals or hopefully ultimately growing the organs in these complex biochambers. That's what I think would be best because then you could use human tissue and you won't have these problems of xenografts. You could maybe even use a person's own stem cells would be really, really cool. But this is kind of where the science sits right now, is we are using these xenografts to a certain degree, and people are trying to get it to work better and better. But ultimately, we usually give out allografts with organ donors. And like I was saying, so autograph, isograph, allograft, xenograph, you're going to increase the risk as you descend. Now, that is another way, of course, the body's immune system is reacting in a clinical setting to something we're putting in. And you're being forced to have this immunodeficiency to accept that. But people can have immunodeficiencies because of their immune cells not working great. And this gets into two different types of immunodeficiencies. These are primary immunodeficiencies and secondary immunodeficiencies. So, primary immunodeficiencies usually end up in a stillbirth or the person who has them sadly making it into uh, usually the latest is the mid to late 20s with these disorders. One example is SCID, severe combined immunodeficiencies. This means something that really, really far up the chain in your immune system does not work. And therefore, maybe you don't have T cells, which would be crazy, right? But in a modern society where we're not exposed as much to a lot of different viruses and parasites and things like that, you can survive a long time with modern medicine. And you might get sick a lot, but you're surviving until mid-adulthood, and then something finally happens. These are genetic origins. They're present from birth, these primary immunodeficiencies, and usually they're going to wipe out an entire arm of the immune system. If it's not so bad that the person is stillborn and is born and able to survive for a little bit of uh, their first few years, they'll usually make it into the teens or the 20s just because we have ways to support their immune system and all of those immune deficiencies they have. But really, it is something that's really hard to deal with. The nice thing about this is genetic engineering allows us to take the genes they're missing and give them back, essentially, and give them these systems back. It's very hard. It is, something that a lot of people are working on, and it's small groups of people that have these immunodeficiencies because generally you don't even make it to birth. And it is it's difficult to address. There's not a lot of money in it, so it's usually going to be academics working with foundations and things like that to slowly move these along. But they are moving along, which is absolutely fantastic. And we are seeing people address these primary immunodeficiencies. Now, there's also secondary immunodeficiencies. These are ones that are acquired later in life. These are things like having HIV turn into AIDS. So that's a virus causing your immune system to just be tired and wiped out. Another way this can happen is chemotherapies, radiation therapies, immunosuppressive drugs, if you're going to be on those for something like transplant. All of these secondary immunodeficiencies are a lot easier for us to address because we see them more, we're able to actually combat them more easily than something like a primary immunodeficiency, which is going to lead to a lot of problems, be hard to diagnose, hard to figure out, and if you just, get the wrong infection, it's kind of a scary ordeal. These, at least we have tests for them. We're chemo and radiation therapy and immunosuppressive drugs, we're giving that to a patient so we can address this secondary immunodeficiency while giving them those treatments. And that makes it easier to control. And these are often treatments that are given to control an underlying disease or that is occurring due to some sort of opportunistic infection like HIV. And luckily, we have things like PrEP for HIV now, and all these put together allows us to really, as clinicians, address these secondary immunodeficiencies. But again, there's tons and tons and tons. Even being on steroids for something like really bad pneumonia, that's technically a secondary immunodeficiency. You're tamping down your inflammation in your lungs, and that is leading you to not cough as much, not risk, having all these secondary issues, but it is a immunodeficiency you're giving to yourself. But you're going to be on something like antibiotics to get rid of the infection during that time. So you're not suffering as much. You are clearing the infection. You are operating normally afterwards, but for that short time, you're having this immunodeficiency. And that's why you're not supposed to do certain things while on steroids. You're not supposed to be on steroids for a long period of time unless you have something that those are addressing as far as an underlying condition. Now, that kind of covers everything that I wanted to talk about here as far as the immune system's interaction really in a clinical setting. And I just want to close with saying that microbiology is such a huge, interesting field. And for me personally, it really shows all of these different things that are at play with us, around us, in us, that impacts our everyday life. From microbiome being very important for mental health to exercise, impacting the type of hormones you release, which impacts how well your immune cells do, which impacts how healthy you are in the day-to-day outside of just the general benefits of being, fitter from exercise. There's all of these cellular responses, which are really interesting. How we've been able to create all these medicines to address all these issues, how we're making vaccines to, deal with viruses like Epstein-Barr virus, which causes MS and causes cancer. And one day we, much like the HPV vaccines preventing all those related cancers. We'll have an EBV vaccine that prevents all of those related cancers and prevents MS. We might one day find out that there's a virus or a parasite or a fungus, something that causes Alzheimer's to be triggered. And we might make a vaccine against that. We might start looking for that when people are doing their yearly screenings and say, hey, you have an antibody to this thing, here's a treatment that we'll give you, and that can help prevent Alzheimer's. So there's a ton of interesting stuff we're doing, including, things like enhanced wound care by impacting the immune cells at the wound site and trying to grow organs or editing pigs' genomes to make hearts more accessible for people that need heart transplants. There's all these cool things going on, but it's all based in microbiology. It's all based in these very early discoveries by people that were living in the 1800s that, didn't even wear gloves and they were, playing around in cows' brains and made us rabies vaccines. Like, there's all this crazy stuff that we can do, and we build and build and build upon the base, the foundation that's been supplied in the times previous to us. And I hope you move on and do some of that building, whether it's having a little bit more knowledge to help patients, whether that's people or animals, or do some primary research, find a new gene in a axolotl that can be put into crops and make the crops better. There's all this interplay and it all is microbiology, and that's why I love it. And I hope after this class you love it just a little bit too. And if you want to talk about any of this, please feel free to message me. I'll talk about this stuff ad nauseum, as I'm sure you've noticed from these videos. So I will cut it off there and wish you the best of luck with the rest of the course material and the final exam and all of that. It's been an absolute pleasure, and I hope you have a absolutely fantastic day.